All right, we're going to go ahead and get started. Good afternoon, everyone, and welcome to the NINDS Heal Pain Therapeutics Development Program webinar. My name is Vakanda Metley. We will start off with a presentation of slides, followed by your questions and answers. Please submit your questions in the Q&A section featured below, and they will be answered at the end of the presentation. This presentation will be recorded and will be placed on our YouTube page in the upcoming days. I will be sending that link along with the presentation slides to everyone that has registered. We ask that you please remain muted for the duration of the presentation, as well as keeping your video off. And I will now turn it over to our moderator for today, Dr. Marianne Palamonter.
Thanks, Rakanda. Hello, everybody, and welcome. Today, I'm going to talk to you about the Heal Pain Therapeutics Development Program, giving you a little background into how it fits into the whole HEAL initiative, and then talking specifically about the PTDP program. Okay. So addressing the unmet medical need for pain therapeutics is the basic goal for the HEAL Helping to End Addiction Long-Term initiative. And the HEAL program takes many different approaches because it's such a complex problem. And so the approach that we're going to focus on today is enhancing pain management. And then the other part of what the HEAL program is about is to improve prevention and treatment of opioid misuse and addiction. So focusing on enhancing pain management, some of the things that the HEAL initiative aims to do is to understand the biological underpinnings of chronic pain, accelerate the discovery and preclinical development of non-addictive pain treatments, advance new non-addictive pain treatments through the clinical pipeline, and inform best practices for effective pain management while minimizing the risk of addiction. So what we'll focus on today, of course, is the second one, which is accelerated discovery and preclinical development of non-addictive pain treatments. So this gives you just a little, if you just focus on the HEAL Therapeutics portion of what the HEAL initiative is doing, you'll see that we have programs that cover a pretty broad spectrum of the early drug development phase. So starting with the discovery portion, we have a grant that's a planning grant it's a short grant meant to support setting up the data, preliminary biology, and mechanism necessary for some of the larger grants. It's called the R61 Planning Grant. And then we have a U19 Team Research Grant, which supports activities like assay development, screening, initial hit characterization, and biomarker development. And then we have the program we'll focus on today, the PTDP program, which supports activities going from hit to lead in small molecule language all the way to the phase 1 clinical trial phase. And then finally, we also have programs from NCATS and the preclinical screening program for pain that support translational assays and preclinical therapeutic profiling. And then on the phase 2 end of the spectrum, we have the EPPIC-Net program. And then you can see at the bottom, we have 10 participating NIH institutes involved in this. And so this is why we can cover virtually all types of pain. Okay, just a little bit on the planning grant. So that planning grant, the RFA number is NS21029. And of course, you'll be having access to these slides at the end of this too. But in any case, the planning grant supports direct costs of $500,000 per year. It's a 2-year duration grant, focuses on building a multidisciplinary team, which you need for any type of drug development project. And that team could then be used in the team research grant, which is what I'm going to talk about next. Finally, another thing about the planning grant is it offers the opportunity to obtain that preliminary data that addresses the entry criteria for the team research grant. The team research grant has a couple major pieces of entry criteria. One is having a multidisciplinary team ready to go and evidence that the team has expertise and can work together. Also that there's a good biological rationale for what you're going to do. And this has to do with the feasibility of your whole idea and your whole project. And it should have support from preliminary data or literature-based evidence. It's a 5-year maximal duration if milestones are met, and it can support direct costs of 1.5 million per year. And then of course, renewals and revisions aren't allowed for either grant. And the next receipt date for both of those is June 9th. And the person who you would contact about these two supporting mechanisms are Julia Bachman. She's the person to talk to. Okay. Then just to say a little bit about one of the other opportunities that we offer as part of HEAL is the preclinical screening program for pain. And this provides a platform to identify and profile non-addictive, non-opioid therapeutics for pain. PSPP takes in biologics, small molecules, natural products, and devices. And what you get out of that is a really nice profiling that allows successful assets to move towards clinical trials. And again, under NINDS direction, rigorous preclinical evaluation of assets can occur in a suite of assays that are performed by contract facilities. So Psychogenics is the contractor for this program, and this is provided free of charge to applicants that are successful in this program. And then the PSPP staff interacts with participant to plan testing, review test results, and discuss future studies. And of course, participant intellectual property and confidentiality are always protected. And this program is open to academic, industry, and government asset owners worldwide. And here your contact is Smriti Iyengar, and you see her information at the bottom of this slide. Okay, then we get to the Pain Therapeutics Development Program, or PTDP. So as I was mentioning, this program supports a wide variety of activities, starting at the hit to lead optimization stage and going all the way into the phase 1 clinical trial stage. So if you're successful in getting one of these grants, it really supports a wide range of early drug development activities, all with one grant. You don't have to keep applying. This supports biologic and small molecule therapeutic development. Another thing about it is that NIH consultants are assigned and tailored to each project based on the needed expertise for the project at that time. And also NIH contract resources are tailored to fit the stage of the project. And the awardee actually has the choice of whether to use these contract resources or not. So NIH, an awardee can choose which NIH contract to use, or they can opt to budget their own contracts in the grant proposal. And then a very important thing about this is that the PI's team and your team and your intellectual property has to be retained by your institution. NIH stays away from any IP claims at all, and so do our contractors. So our main goal is to make sure that stays with you. And then just on the right side of this slide, to give you an idea of how things work if you're funded for one of these grants, is that if you're funded, a lead development team is formed around your team. And this team consists of you and your team, industry seasoned consultants, and NIH staff. The consultants, as we mentioned before, are assigned based on where you are in your drug discovery and development project. And these consultants have expertise in assay development, med chem, chemistry, manufacturing controls, PK, DMPK, and pharmaceutical development. And then the NIH staff largely plays 2 roles. You'll have an NIH staff project manager, and then you also have NIH staff subject matter experts in pain. And then below this shows you some of the vendors that are our contractors, if you opt to use them. So for medicinal chemistry, we have Curia as our contractor. For PK and Tox, we have SRI, TRL Labs, ITRI. And that's it for now. And then we have a data management contractor that is collective data discovery, and that's CDD. And then for manufacturing and formulation, we have Curia and MRI Global on the small molecule end, but then as I mentioned, we support biologics development too. So ABL, Battelle, and Leidos also do manufacturing and formulation for biologics. And some of their contractors also do toxicology and DMPK. For clinical trials, we have two vendors, Duke University and PPD. The next receipt date's June 15th of this year for this particular opportunity. I thought we'd show you an idea of how the pain therapeutics development program portfolio looks now. So it's been in existence. This shows data representing the application, the awards that we funded since 2019 when the program started. So you can see that we have right now 15 projects have been awarded to date. And from those 15 projects, you can see that there's a pretty wide variety of targets, biological targets that have been chosen. And you can see that most of the therapeutics are small molecules, roughly like 60 or 70%. And then the remainder is biologics. But that also means there are quite a few biologic awards that have been made as well. And then you can see from the map that institutions that have been given these awards are really across the entire country. Unfortunately, though, mostly just hitting the East Coast and southern states. We still need to get in probably more Midwestern and northern states. And then if you look at the pain conditions that are represented by these 15 projects, the majority have been for neuropathic pain, but we also have quite a few projects that are looking at chronic pain. And then we have knee pain, osteoarthritis, diabetic neuropathy, complex regional pain syndrome, sickle cell, and so on. So we have a number of pain conditions. And as I mentioned, because so many institutes are signed on to this funding opportunity, most types of pain are covered through this funding opportunity. Here are some of the features of this, and the funding opportunity RFA is NS 21010. So some of the features of the grant itself are that it's phased and it's a milestone-driven cooperative agreement type of grant. It supports, as we mentioned, hit to lead activities, supports lead optimization, selection and characterization, biomarker optimization, PK/PD development, and also IND enabling studies in phase 1 trials. And again, this is just some activities that they support. But I think the main thing to remember too is this particular funding opportunity is set up to support the process of developing a biologic or a small molecule therapeutic from early stages all the way through phase 1. And so that's how our infrastructure is put together. The end goals and milestones are things like identifying and fully characterizing the lead therapeutic, identifying a target engagement biomarker, seeking partnerships, completing IND enabling studies, filing an IND, completing phase 1 trials. And then the big thing is that hopefully once you would make it through this particular program, your project would be ready for phase 2. Okay. So one other thing to point out about this grant is that applications can enter in discovery or development stages. But the important thing to remember is that it is a phased grant. So it has a UG3 phase and a UH3 phase. All projects have to begin with a preparatory UG3 phase. And that UG3 phase can be up to 2 years in duration. And then the UH3 phase is usually the remainder of a 5-year grant, so the remainder would be 3 years. So as I mentioned, people can enter in discovery if they can complete entry criteria for hit selection. They can enter in various stages. We've had people enter in the very exploratory stages. We've had them enter in hit to lead, and even when they're ready to start lead optimization. And then we've had people enter the development stage. Usually that's when they're in the stage of beginning manufacturing, doing their dose range finding studies, things like that, and then moving through phase 1. And so again, in an application, it's important to remember you must include both the UG3 and UH3 plans in the application, as well as their budgets. So sometimes we have people, I think, who think that you're only applying for the UG3 phase right now. That is not true. So you're applying for both the UG3 and the UH3 phase. When you transition, we'll talk more about transition in a few minutes, but the transition is an administrative transition only. It's not another peer review. Okay. And then who's eligible? Academic institutions, small businesses, foreign institutions, and components are also eligible. All of these are. Now, a little aside, if the application's from a foreign organization, reviewers will assess whether the project presents special opportunities for furthering research programs through the use of unusual talent, unusual resources, populations that are unique, environmental conditions unique. In other words, it has to be something that you could argue is just not readily available or accessible in the United States. Okay, so let me talk a little bit about the entry criteria. So for the discovery stage, if that's how you're entering this, you need to have a rigorous biological rationale for the intended approach that you're going to propose. Should have a promising small molecule or biologic starting point for optimization. And you should have scientifically sound assays to optimize and test the agent. And so when we're saying scientifically sound, we mean that you've done some work to validate them, even if it's just in terms of how reliable are the assays. Are they sensitive enough to detect what you need to detect routinely? And is your dynamic range sufficient to actually tell you the picture of what the agent's doing? And then entry criteria, if you're coming in at the development stage, there you must show that you have identified a candidate therapeutic. And so along with that, you should have in your application a strong data package linking the modulation of the therapeutic target by the candidate therapeutic to disease modification. You should also have biological activity and ADMET properties listed, and you should be able to show that they're appropriate for the intended use. So, you know, as like most grants, there are some activities that are not responsive. And that means that they, you know, you can send it in and it can just be sent back to you and it won't be reviewed. So just make sure you're looking at carefully at the funding opportunity announcement so you know what's non-responsive. So this list is pretty much what we have in the FOA funding opportunity announcement. And so things like screening for initial hit identification, that's too early. So that would be something that might be done in the U19 that I talked about earlier, for example, but that is not appropriate for this particular grant. Now, I should point out that the key word here is initial hit identification. We understand that as you're trying to optimize and trying to choose your lead, you're going to continue screening, of course, but this is to find your initial hits, that you should have some initial hits when you come in. And then another non-responsive thing is basic research and studies of disease mechanism for disease mechanism's sake. Animal model development is not responsive. The development of diagnostics and diagnostic devices is not responsive. Clinical studies beyond phase 1, same thing, not responsive. And then pay attention to this, opioid sparing projects and projects targeting the mu opioid receptor are also not responsive, because our whole idea is to come up with a non-addictive therapeutic. So here are some typical activities for this 2-phase grant mechanism. So for the preparatory or UG3 phase, these are some of the things that a lot of applicants that are successful come in with. They're optimizing using potency and efficacy screens, preliminary efficacy and tox testing and appropriate animal models for pain, characterization and testing for in vitro ADME. And of course, ADME means absorption, distribution, metabolism, and excretion. And also things like initial optimization of pharmacodynamic or target engagement biomarkers associated with the therapeutic target or pathway. And then for the execution or UH3 phase, some of the activities that are typical are any further optimization as listed in the preparatory phase if needed, because this is a continuation, of course. And then pharmacokinetics, biodistribution, gene expression, tumorigenicity, and immunogenicity are all things you might do in the UH3 phase. Same way with cell bank development and testing, formulation and manufacturing for toxicology and human clinical studies, then IND enabling characterization of the clinical candidate, non-GLP and GLP tox, and then IND submission and phase 1 clinical testing, all typical of the UH3 phase. Now, I mentioned before that transitioning from the UG3 to UH3 phase does not require another peer-reviewed type of application. So instead, it's an administrative review. So at the end of the UG3 phase, NIH program staff and leadership will determine if the project can advance to UH3. NIH program staff and leadership also conduct annual administrative reviews. So I think that's something important to understand, is that the main transition review is at the end of the UG3 phase, often after 2 years, if that's how you set up your application. And that's, you know, there will be, as a program staff are involved in the decision making, and program leadership are as well. But the thing is that you're not totally out of the risk level there, because then we do conduct the annual administrative reviews also, just to make sure that as we move into more expensive things that we're funding, like manufacturing and toxicology, for instance, that the project still looks feasible. Reviews are based on successful achievement of milestones, the overall feasibility of project management, including biomarker work, and considering data that may not have been captured in milestones. Reviews are also based on competitive landscape for the disease indication and drug target. And remember, the competitive landscape can change. And then reviews are based on program priorities and, of course, availability of funds. So let's talk a little bit about milestones. So milestones are a really important component of that administrative decision to transition to UH3. Milestones should be included in the grant application. They're used for measuring success in achieving each of the research plan's key objectives. One or more milestones should be used for each key objective. And so you should also have milestones for each year. For each milestone, provide details on the methods, assumptions, and experimental designs and data analysis plans that they're based on. Specify quantitative criteria for measuring success. Because what we're trying to find out really is, as you're moving from year to year, where are the showstoppers? What do you absolutely need to show in order to convince yourselves that the project should be continued and that it can be continued? So quantitative criteria should be robust and consistent with the state of the art in the field. Quantitative criteria for success will be used for making go, no go decisions, and this should be specified. You should also specify the timeline for each milestone. It helps us if you have a timeline where your milestones are, mapped onto that. And then milestones, another thing that is important to point out is that you will propose these milestones in your application and they will be reviewed. But then if you get an award, probably NIH staff and the lead development team that is assigned for you will probably further refine these milestones, because after enough discussion goes on, you might find some of the milestones may not have been as relevant as you thought. We want to make sure things are realistic and achievable, if possible. Okay. Then the other thing, just a little aside on intellectual property expectations. So their IP is based on some of these tenets, that inventorship is determined per US patent law. So whatever US patent law says, that's the definition of, like who is going to be the inventor. The NIH Pain Therapeutics Development Program has absolutely no stake in intellectual property. Again, we want that to be yours. Prior to grant award, the PI's institution must have upfront IP agreements in place with all potential inventors. These agreements must address who will hold the title to the IP on new chemical matter in use or a new biologic, and they should also address royalty arrangements. IP agreements should aim for unencumbered intellectual property, which is consistent with the whole PTDP goal, which is to create a licensable product that can be eventually used by patients. Here are a few little tips. Please ensure that your application has separate budgets for each year of the grant. So a grant that's for 5 years should have, separate budgets for each of the 5 years. So there should be 5 years of budgets, plus the cumulative budget at the end. And you should also make sure if you're going to have subawards or subcontractors that you're going to ask funding for, that's also indicated in your budget plan. And there should be also in your application a table in which activities that are conducted by your team and associated personnel, and for that reason are funded by the UG3 UH3 grant. And it should indicate that, and it should also indicate which activities you want the NIH contract research organizations or consultants to do. So we need to know what activities you're going to do and that you have put into your budget, and which activities you want us to do through our CROs. And then another thing that's important in the application is a table of proposed activities that provide the following types of information: what the activity is, what the throughput of the activity is, and where it's going to be done, and what are the advancement criteria. It's almost as if you're writing a table that is a summary of your screening funnels. You should also include a target product profile table, and that's more facing the end of what it's really reminding you of your goals. So it's saying clinically what is ideal and what is acceptable in terms of the final route of administration you're going to have, the patient population you want, the formulation that you're going to have, things like that. You should also address the biomarker component in your application. You don't necessarily have to develop a biomarker from scratch in this, but if at all possible, you should have some sort of biomarker that you can measure for PK/PD relationships, for instance. But we do realize that if you have to develop a biomarker completely from the top, that might not be possible to do along with, like developing your therapeutic. And so this is about developing therapeutics, not developing biomarkers. So we just want to make sure that you think about the biomarker and address it in your application. But if it looks like there are going to be issues with what you're addressing, point those out in your application, and point out potential other ways to get PK/PD. And then another thing that's important is addressing intellectual property in the other attachments section. That does not go into the research strategy section. And then if you're asking for NIH to conduct the clinical trial under our contracts, then mark your application clinical trial no. And if you're going to do the clinical trial on your own, it is highly likely that you would be marking it as a delayed onset clinical trial. Okay, and then this is just to, in some of our older versions of our RFA, and I think even in the beginning of this one, we still required cost matching, but we do not anymore. So just make sure you notice that when you look at the FOA or the NOFO, that will point out this notice, which says you don't have to do that anymore. Okay, here are some things to do. Read the funding opportunity announcement carefully. Definitely discuss your proposal with us, with the program director. So, you take a look at who is listed as a program director for this, and usually it's me. So just do call me if you have any type of question at all. I don't mind talking to you. It's much, much better if we make sure we're all on the same page before you spend a lot of time writing an application that doesn't necessarily fit our infrastructure or our goals. So just remember that, contact us. We're happy to talk to you. Stick to the page limit, especially for your research strategy. I think it's 12 pages. Make sure that you have a well thought out scientific premise. And it is important to discuss the quality of the data presented in prior publications in a detailed manner. So if, to back up your premise, you may have your own preliminary data and you may have data from prior publications. Again, make sure that you address how reliable that is. And that gets to the whole rigor idea. So, make sure controls that are being used for each type of experiment are the correct ones, and highlight potential confounds like surgery exposure, genotype, and so on. Include details within the experimental design about the reduction of potential bias, including the use of blinding randomization and inclusion-exclusion criteria. And then finally, describe the source of the data on which the sample size estimation is based and details about the analysis itself. So it always is helpful to have a statistician that you're working with. Some additional things to do is to complete your required registrations like SAM at least 6 to 8 weeks in advance of the receipt dates. Consider submitting your application early. This gives you a chance to react to issues that might result in your application being withdrawn. So submitting it early and talking to us is a good combination. Discuss your budget with NIH staff if you have questions. Talk with your own tech transfer and business development group. Just make sure that you've thought through the whole idea of planning for funding patents and licensing activities and making sure that you have permission to use reagents, animals, things like that. Things not to do. Do not plan for this to be the sole funding in your lab because, as we've mentioned, this is milestone driven and these programs can end abruptly if you do not meet your milestones. Remember, at the end of each year, this is looked at. Do not forget to address the use of NIH contract resources. When you think about using these resources, remember that our NIH contracts are not set up to look at disease biology. The only thing that does that is the preclinical screening program for pain. And you do need to apply to that. That's separate from the grant, but it's absolutely, it works very well if you apply to that and plan to use it while you're working on this grant. And keep in mind, that's free of charge. Please do not include NIH contracts in the application budget because we are the sponsors for those, and those are also free of charge to you. Do not reach out to planned NIH contractors for letters of support because that has nothing to do with your budget since you're not paying for them. Okay. All right, so that's it. Questions. And here's all our contact information. So I'm the program lead. Chuck Sywin is my boss. He's the director of small molecule neurotherapeutic development. Carolyn Bonder is the health program specialist who does so much of the coordination and type work in this program. And then Rakonda Medley, who is our operations coordinator and who is managing the Q&A. All right, and then make sure to visit PTDP online with this QR code. Okay, questions?
Thanks, Marianne. I just wanted to remind everyone to please use the Q&A section below and that the slides in the presentations will be sent out in the near future. Thank you.
Okay, so the first question is, can you elaborate on what is meant by opioid sparing projects? Would this include a project that reduces the amount of opioid required to produce analgesia?
And that is correct, it would, yes. I don't think there's much more to say about that either, because that's just the way it was set up. We really want to make sure we're not getting in new opioid targets.
My project is already ready for IND enabling studies. Do I still need to include the preparatory phase in my grant?
So okay, so you're coming, I guess I need a little more information about that. If you're ready for IND enabling studies, does that mean you've chosen the lead and you've started characterizing your candidate therapeutic?
Well,
I guess,
I think it means, can you skip the UG3?
yeah, so that you cannot. You would be doing preparatory activities for that particular stage you're in. So preparatory activities for someone coming in the development stage, which is what you would be things like actually doing your dose ranging PK, your initial manufacturing characterization work of your therapeutic, that kind of thing. And you could, I mean, you can always propose a 1-year UG3.
Great. Can you elaborate on the preclinical screening program for pain you just mentioned?
Right. So as I mentioned, the work is done at Psychogenics. It's a profiling type of program where, first of all, the asset that you have that comes in, if you get accepted into this program, then your asset gets run through some in vitro pharmacology screens to assure that you're not hitting new receptors. And then it also gets run through a pharmacokinetics profiling as well. And then once it goes through that, then it's you run through various pain types, like I'm not exactly sure what all the assays are in it right now, but you first look at the general type of pain, acute pain, chronic pain. And then once that looks positive in that, then you move on to a specific pain indication of profiling. And they also will look at liability, like self-administration, for instance, to make sure that you're actually not having an addictive propensity.
Okay, so I think this question will get at a little more granularity on the UG3, UH3. So they have initial hits from a primary screen where they need to test potency, efficacy, selectivity. Is it right that they apply for UG3?
Right. So again, you have to apply. UG3 has to be in every single application, no matter where you are. So, again, we call it preparatory, but that just means, we want to make sure that you are where you think you are in terms of the phase of drug discovery you're in. So even if you're ready ready to start doing dose range finding studies, for instance, we want to make sure that you really have characterized your lead appropriately and that there aren't any holes, because then we'll help you fill in those holes first. And, if you're in the discovery stage and you have a good set of hits, you have efficacy and so on, but you still need to optimize, well, that can be the UG3 phase too. So you just have to remember, everyone has to do a UG3 phase.
Are there budget limits on the UG3 or UH3 phase?
No, there aren't any specific budget limits. Your budget just has to be well justified by the activities you propose. And this brings up to the importance of really doing your homework about the different activities that occur in drug discovery and development. Make sure you're realistic with your budgets and make sure you've really thought through what you need. And then, we're just looking for, we're certainly our reviewers, and we are going to spot things that are unrealistically high, but we also sometimes spot things that are too low. So we just want to make sure you can do what you're saying you're going to do.